the role of the gut microbiome and its metabolic products on the maintenance of muscle mass, muscle composition, essentially how much fat is within muscle, which is not good for muscle health, and on physical function in older adults. So this is a picture from a review in 2016 that essentially shows that the gut microbiome has been implicated in the health and functioning of almost every tissue or organ in the body. So starting from the brain, it's been linked with autism, stress, and stroke. With the lung, in terms of allergic asthma, it's been linked with the liver, with non-alcoholic fatty liver disease. It's been linked with the skin, atopic dermatitis. It's been linked with fat, adipose tissue in terms of inflammation and obesity. And in terms of the whole body, it's been linked with type 2 diabetes, lupus, malnourishment, and atherosclerosis. Notably absent from this review, as recently as 4 years ago, is muscle. So obviously that's what I'm going to talk about today. So, so today I'm going to go over, so we reviewed the existing literature in 2018, our group, what was known at that time. So I'm going to talk about that, a lot of that data. And then I updated the literature based on, I did a lit review. There were about 9 studies that came out last year. It was a big year for the role of the gut microbiome on muscle last year. So I updated that in a review from last year. And then what are my contributions to the gut muscle axis? So I'm going to talk about a couple studies that our group published in the last 6 months. So first, let's even see if there's enough data to define a gut muscle axis. So what we're looking at here is SPF. These are specific pathogen-free mice. These are mice that have a microbiome, a defined microbiome, in comparison with germ-free mice, mice that don't have a microbiome. They're grown in a bubble. And what I highlighted is that muscle in germ-free mice is lower. There is less muscle mass in the germ-free mice compared to the microbiome-containing mice. Now, it's important to also note that body weight was not different when comparing these 2 groups of mice, because if body weight was down and muscle was down and liver weight was down, is it really an effect of the microbiome on muscle, or is it just an overall effect of the smaller body, smaller animal? So this group here compared the muscle mass to body weight ratio, and it was still lower in the germ-free mice, which suggests a role for the gut microbiome on the maintenance of muscle mass. So these data were confirmed in a study last year. And again, what we're looking at is the muscle weight to body weight ratio. And they looked at various lower leg muscles, soleus, gastrocnemius, tibialis anterior, TA, quadriceps. And they compared pathogen-free mice, again, these are mice that have a microbiome, with germ-free mice. So let's start there. So for the gastrocnemius, tibialis anterior, and the quadriceps, the germ-free mice had lower muscle mass normalized to body weight when compared with the microbiome-containing mice. So what happens if you colonize germ-free mice with fecal samples from aged, matched microbiome-containing mice? And what happens to muscle mass? So starting with the gastrocnemius, we see a trend for an increase, but the muscle mass to body weight ratio was increased for animals that germ-free mice that received the fecal transplant from microbiome-containing mice, and also for the quadriceps, it was increased. So collectively, these data suggest that there are factors within the microbiome that promote muscle mass. So the microbiome isn't just comprised of bacteria, it's also comprised of viruses, fungi, archaea, etc. So evidence that it's bacteria that may be involved in mechanisms related to muscle mass comes from studies of antibiotic treatment in mice. And in looking at these data here, starting from control animals and then antibiotic-treated animals, we can see that the antibiotic-treated animals have a lower muscle mass to body weight ratio for 2 of the biggest muscles in the lower leg, gastrocnemius and quadriceps. So in further support that gut bacteria play some role on the maintenance of muscle mass, when they remove the antibiotic treatment and then expose those mice to bedding and feces from mice that had not been treated with antibiotics, this allows the microbiome to reseed in the antibiotic-treated mice. So what happened to muscle mass? And what we can see is that it increased in both muscles. So these data have been confirmed in other studies, at least part of it in terms of antibiotic treatment, where antibiotic treatment results in a lower muscle mass to body weight ratio in a couple other studies. So collectively, these 5 studies demonstrate that gut bacteria may be involved in mechanisms related to the maintenance of muscle mass. So that doesn't say anything about which gut bacterial species may be involved in mechanisms related to muscle mass. So what about those? So in this study from last year, the NEES group compared young animals and then what they considered old, even though their definition of old were 10-month C57 black 6 mice, which is not at all old. Average lifespan is 30 months. Nonetheless, they call them old. It's not old, but so they fed these 10-month-old mice, black bars, either Lactobacillus casei or Bifidobacterium longum, and then looked at the muscle weight to body weight ratio for the quadriceps, gastrocnemius, and tibialis muscles in the lower leg. And hashtags were where they saw significant differences. So Lactobacillus and Bifidobacterium increased muscle mass compared to untreated mice, and Bifidobacterium increased tibialis muscle mass when compared with untreated mice. So separately, in a leukemia mouse model from a longer time ago, 8 years ago, so they so muscle mass is reduced in this leukemia mouse model, and when they supplemented the mice with various lactobacillus species, muscle mass was increased. So let's have a look at that data. So this is the tibialis muscle mass, lower leg muscle, and just weight. They didn't look at the body weight, the muscle mass to body weight ratio. So first, starting with the controls, tibialis weight, controls plus the lactobacillus bacteria, no effect on muscle mass in the controls. Leukemia mouse model had a small but significantly different or reduced muscle mass compared to control mice. And then when they fed the leukemia mouse model, the probiotic species, the lactobacillus species, there was a small but significant increase in muscle mass. So while these data suggest a role for the gut microbiome, gut bacteria, and individual gut bacterial species on muscle mass, it doesn't say how that they could do that. So let's take a look at how this could possibly happen. So metabolites produced by gut bacteria can affect muscle mass. And one group of metabolites that can do that are the short chain fatty acids, which are produced by the bacterial fermentation of dietary fiber, which have been shown to increase muscle mass. So that's what's shown here. So in a study from last year, they looked at the gastrocnemius muscle weight divided by body weight in germ-free mice, and then germ-free mice that were fed short chain fatty acids. So it's important to note germ-free mice don't have any short chain fatty acids in their feces because they don't have a microbiome to produce them. They can't degrade dietary fiber to produce short chain fatty acids. So when you give germ-free mice short chain fatty acids, their gastrocnemius, their muscle mass divided by their body weight is increased. And just to highlight, these are the short chain fatty acids that they gave them were acetate, propionate, and butyrate. These are 2-carbon, 3-carbon, and 4-carbon fatty acids, hence the name short chain fatty acids. So going into old mice, in this study, they gave butyrate, only one of these short chain fatty acids, starting at 16 months of age until 26 months of age. 26 months of age is pretty old in a mouse. Like I said earlier, the average lifespan in C56 black 6 mice is 30 months. So 26 months is relatively old. So first, we can see that there is an age-related decrease in muscle mass of the gastrocnemius, soleus, tibialis, anterior, and quadriceps, again, lower leg muscles in the mouse. So white bar to black bar, it's decreased in each of these muscles, indicated by the stars. So what happens when they fed butyrate? So young mice fed butyrate compared with unsupplemented young mice, no effect in any of the muscles for the young mice. But when they fed the older mice, starting at 16 months of age for 10 months, butyrate, when they fed them butyrate, each of these muscles, the muscle mass to body weight ratio was increased. So hashtag, 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 which again supports a role for these short chain fatty acids on the maintenance of muscle mass, both in young and old animals. Now, whereas there are metabolites that positively affect muscle or can positively affect muscle in animal models, conversely, there are also gut bacteria-derived metabolites that can negatively affect muscle. Now, one of these is called indoxyl sulfate. Indoxyl sulfate is found in the blood from microbiome-containing mice, but it's not found in germ-free mice, thereby identifying it as a gut bacteria-derived metabolite. So in vitro, indoxyl sulfate negatively affects muscle development. Muscle development proceeds from satellite cells, which differentiate into myoblasts, which undergo further differentiation and proliferation into myotubes, which then undergo several cycles of fusion to form mature muscle. Indoxyl sulfate negatively affects each of these components. So first, In the presence of indoxyl sulfate, when controlled with untreated myoblasts, there is less myoblast proliferation in the presence of indoxyl sulfate. So, then going forward, when myotubes are exposed to indoxyl sulfate, they shrink, they atrophy, and there are also less myotubes in the presence of indoxyl sulfate. So at least two of these components of the road from satellite cells to mature muscle are negatively impacted by indoxyl sulfate. So what about the impact of indoxyl sulfate on mature muscle? So indoxyl sulfate is a uremic metabolite, meaning that its levels are elevated in the presence of kidney disease. So to simulate that condition, the authors of this study removed the kidney. So these are animals that have only one kidney, and then they injected indoxyl sulfate daily for 12 weeks, and then looked at various outcomes. And what we can see is that in indoxyl sulfate treated mice, there was a reduction in muscle mass for the tibialis anterior, for the soleus, and the gastrocnemius. Each of the muscles had decreased muscle mass. But I should also point out that body weight was also decreased as a result of the indoxyl sulfate injections. So is this an effect on muscle mass, or is this a bigger effect on body size? That's not known, and studies haven't replicated the in vivo effects, but hopefully at some point in the future I can go after studies related to that. So in addition to indoxyl sulfate, another uremic metabolite, this time known as paracresol sulfate, can negatively affect skeletal muscle. And again, skeletal muscle is not found in conventionally raised microbiome-containing mice, or it is found in conventionally raised microbiome-containing mice. It's not found in germ-free mice, thereby identifying it as a gut microbiome-derived metabolite. So exposure to paracresol sulfate negatively affects muscle composition, in this case, indicated by tissue lipid content. So mice that were injected with paracresol sulfate once a day or twice a day, something like once or twice, I can't remember, but for a month, had increased levels of lipids within muscle. And it's important to note that they also had a mouse model of chronic kidney disease, which is a situation where there's also an increase in muscle, gastrocnemius muscle, tissue lipid content. And just by itself, paracresol sulfate was almost able to replicate the adverse effects on muscle composition compared to the more extreme case, chronic kidney disease. So just as a quick intro, why is muscle composition important? Well, in older adults, poor muscle composition, in this case indicated by increased lipids within and or between skeletal muscle cells, it's associated with insulin resistance, reduced mobility and physical function, frailty, and an increased all-cause mortality risk. So collectively, all the data that I've shown so far suggests a support role for gut microbiome-derived metabolites on muscle mass and composition. So what about the role of the gut microbiome on muscle function? So physical function is reduced in both germ-free and in antibiotic-treated mice. So first, in germ-free mice compared with microbiome-containing mice, there's decreased grip strength. And then germ-free mice compared with microbiome-containing mice have a reduced swimming endurance capacity. That's what's shown here in both of these two studies. And then when you treat microbiome-containing mice with antibiotics, an intervention that reduces gut bacterial content, we can see that half of the colony or half of the cohort that was treated with antibiotics is exhausted at about 100 minutes, where the non-antibiotic treated mice exhaust much later at about 150 minutes. So antibiotic treatment negatively affects treadmill endurance capacity. So again, this suggests a role for the microbiome on physical function, but it doesn't tell us about gut bacterial species that can affect muscle strength or endurance capacity. So which could those be? So similar to the study that I mentioned earlier, where they called 10-month-old mice old, which isn't. Again, they fed Lactobacillus casei or Bifidobacterium longum, and they also saw increases in grip strength compared with unsupplemented mice. So we see the Lactobacillus and Bifidobacteria had higher grip strength compared with unsupplemented controls. And then swimming endurance capacity in the germ-free mice was reduced compared to mice that have a microbiome, but then feeding germ-free mice Bacteroides fragilis increased their swimming endurance capacity. In another study, so when looking at four separate groups of germ-free mice, pre-supplementation with various bacteria, and then after supplementation with various bacteria, Eubacterium rectali, shown here, and Clostridium cocoides, shown here, had higher swimming endurance capacity when compared with germ-free mice that were not supplemented with bacteria. And then in another study published last year, they gavaged Veillonella atypica into mice that actually had a microbiome. So they increased the amount of Veillonella bacteria that were in the mice, and they saw an increase in treadmill endurance capacity. So similar to what I showed earlier, while that suggests a role for individual gut bacterial species on physical function, it doesn't say anything about how these gut bacteria can affect function. So what about bacterial factors that can affect physical function? So once again, the short-chain fatty acids are involved. So whereas they improve muscle mass, they also improve muscle strength. So we see that here, germ-free mice treated with short-chain fatty acids have a higher grip strength when compared with germ-free mice. And then interestingly, so it's important to note that the germ-free mice that got short-chain fatty acids, these were a combination of acetate, propionate, and butyrate, which may be important because when fiber is fermented, it isn't fermented into only butyrate, it's fermented into proportions of each of these short-chain fatty acids. Now, why that's important is because when aged mice were fed butyrate for 10 months, there was no difference in grip strength. So here we have the control diet, young versus old. There's an age-related decrease in grip strength. And then young versus old that were treated or supplemented with butyrate for 10 months. Again, there's that age-related decrease in grip strength that was not attenuated by butyrate supplementation. So it may be that feeding short-chain fatty acids or feeding fiber, it may be important to have all of the combinations of these short chains rather than individually like butyrate in this case. So in other studies, acetate, the other short-chain fatty acids, acetate and propionate improve treadmill endurance capacity. So here, as I mentioned, antibiotic-treated mice in green run a significantly shorter distance on the treadmill when compared with untreated control mice. So when you feed antibiotic-treated mice, the short-chain fatty acid acetate is shown here, they have better treadmill endurance capacity. And then if intrarectal infusion of propionate, which is where it would be produced when it's fermented by dietary fiber. So propionate in the colon resulted in increased treadmill time in mice, which is shown here versus injection with PBS. So collectively, these data support a role for the gut microbiome, individual bacterial species, and bacterial metabolites on the maintenance of muscle mass and physical function, but in mice. So what about in humans? So there isn't a lot of data in humans, and I'm just gonna kind of highlight a couple key points in the interest of time. So first, elite athletes have a different microbiome and higher fecal short-chain fatty acids when compared with normal weight and obese controls. So what we see here is the unweighted unifrac. So that's essentially a measure of microbiome community structure. And what we can see is that the athletes in the black dots cluster separately. I mean, it's pretty clear they cluster separately from people who had a BMI less than 25 and higher than 30, as shown here. And then as you would expect based on the animal data, the athletes have higher levels of fecal short-chain fatty acids, as shown here. So acetate, propionate, and butyrate, gray bars, higher when compared with the less than 25 and higher than 30 BMI controls. So athlete, 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 higher than the green controls. So what about in older adults? So the first study in this field was 15 years ago. Van Tongeren, his group, looked in a small study, looked at differences for various gut bacteria when comparing less frail older adults. These were about 88 years old, less frail older adults with more frail older adults. And what they found was were differences for 4 main groups. Bacteroides prevotella. Prevotella can produce short-chain fatty acid propionate. Fecalibacterium prausnitzii, and Lactobacillus enterococcus. These 3 groups of bacteria were higher in the less frail older adults when compared with the more frail older adults. And just to highlight, Fecalibacterium are known butyrate producers. Again, Prevotella are known propionate producers. And then if you remember in some of the earlier slides, Lactobacillus species were involved in mechanisms related to muscle mass and muscle function. So I'd argue these bacteria may be involved also in mechanisms related to at least frailty in older adults. Now, what I didn't talk about yet were enterobacteria, and their importance is illustrated as they were higher in the more frail group and lower in the less frail group. And these data have actually been shown in other studies where enterobacteria are higher and more frail. I left an extra in there, sorry about that. enterobacteria higher and more frail when compared with less frail older adults. And they'll pop up again in a few slides. So it's important to mention that frailty was measured in these studies with a questionnaire that included questions about mobility and physical fitness, but it wasn't specific for physical function or muscle mass. So are there studies that more specifically address the gut muscle axis in older adults? So that's where my contribution starts to come into this story. I didn't actually have interest in the gut microbiome at all in terms of muscle. It kind of evolved out of my findings in a few studies, 2013, 2014, and 2015. So what we did is we used a serum metabolomic approach. So looking at about 300 to 400 different metabolites in blood and how they associated with muscle mass, muscle composition, and physical function in older adults, actually in both young and older adults. And interestingly, I've listed these metabolites here, which are either exclusively derived from gut bacteria or are products of gut bacteria human co-metabolism. And these metabolites were associated with muscle in both young and older adults. And just to highlight real quickly, if you remember, indoxyl sulfate and pericresol sulfate were negatively associated with muscle in animal models. Similarly, they were also negatively associated with muscle-related outcomes in older adults. So with these data and other published data, I got a few grants published, and with that in mind, started to investigate this question. So my hypothesis was that gut bacteria are associated with and causally involved in mechanisms related to the maintenance of whole body lean mass and physical function in older adults. And we published these data last year, and it was a relatively small study. Two different groups of older adults that we defined as high functioning, HF, and low functioning, LF. And our main inclusion criteria was the short physical performance battery, SPBB, which includes a few measures of physical function. So we expected to have two groups that were different in terms of physical function, and in addition, that were potentially different in terms of body composition, and that's what we ended up with. So just real quick, these two groups, high functioning and low functioning, were not different in terms of percentage female, age, and BMI when comparing the two groups. The high functioning group had a more favorable body composition, including 5% higher whole body lean mass percentage and also 5% lower fat mass percentage, but no difference in bone mineral content, BMC. And as expected, there was better physical function in the high functioning group, whether defined by the SPBB and each of its components. These high functioning older adults were able to complete a 400-meter walk faster than the low functioning group, and the high functioning group also had better muscle strength as indicated by the leg press 1 repetition maximum. So the obvious question is gut microbiome composition different when comparing these two groups? So what we're looking at here is average family level relative abundance when comparing the low functioning group with the high functioning group. And what we can see is that the high functioning group has more Prevotalaceae when compared with the low functioning group. Now, conversely, the high functioning group has less enterobacteria, whereas the low functioning group has more, about 3-fold more. So if you remember earlier in the Van Tongeren study, they actually saw similar findings where more enterobacteria were associated with frailty and more Prevotella were found in less frail older adults. So we kind of see a similar pattern here in our our cohort wasn't frail. So these data are interesting, but are they reproducible? And so if I would go and get my body composition, my lean mass, my fat mass by DEXA or CT or some other means measured, and my physical function also measured, today, and then didn't change my diet or my fitness approach or my supplements or whatever, sleep, all the factors, and then remeasured a month from now, we'd expect that those data would be pretty similar. But in terms of gut microbiome studies, that it isn't so well studied for people to look at, for groups to look at, are your findings reproducible? Do you see similar effects longitudinally? So we wanted to investigate that. So 1 month after the baseline visit, we had our subjects come back and then perform the measures of physical function and body composition. And we also looked at their gut microbiome composition. And if we would see changes in the gut microbiome at the baseline visit that were similarly represented at the follow-up visit, this may add additional strength to our findings. So once again, we saw that Prevotella were higher in the high function group. You see that here. And it looks like the enterobacteria would be higher in the less functioning group, lower functioning group, but those data weren't statistically significant. And if you remember that we had a total study sample size of 29 subjects, it's possible we weren't powered to see this as a significant difference. And then I haven't shown all the data, for differences in bacteria, but these were the main findings. But we also saw differences in genus level Privitella and Barnesiella and the bacterial species Barnesiella intestinal hominis. These bacteria were significantly different at both study visits. So based on that, I'd argue that those bacteria may be involved in mechanisms related to the maintenance of muscle mass and physical function in older adults. So those are associations though. How can we test causation? So one way that people have done that is by fecal transfer into germ-free mice. So we took 6 fecal samples from the high functioning group and 6 from the low functioning group, transplanted them into young germ-free mice, the animals that don't have a microbiome, waited a month, and then looked to see if we had high functioning and low functioning mice respectively that correspond with their respective donors. So the first step is, do we see microbiome differences when comparing the human fecal donors and their colonized mice? And then do we even see differences in body composition or physical function in the human colonized mice? So first, I'm looking at microbiome differences between the 12 human fecal donors, the 6 high functioning and 6 low functioning human fecal donors, and their microbiome containing mice. These are bacterial differences at each taxonomic level, so phyla, class, order, family, genus, species, and I'm not going to go over all of them, but I'd predict that the strongest data for a role for the gut microbiome on the maintenance of body composition, including lean mass and fat mass and physical function, would be present for the findings that we saw in our larger human group, N equals 29, our smaller human group, N equals 12, the human fecal donors, and then if those data were also significantly different when comparing the human microbiome containing mice. So for that, we see similar differences for each of those 3 groups, for Privitellaceae, Barnesiella, Privitella, and Barnesiella intestinal hominis. So now the key question is, did we see changes in body composition or physical function? Because if we just saw changes in the microbiome, but didn't see changes in outcome measures, it doesn't really mean much. So I should have mentioned that we used 3 mice per each human fecal donor. So each fecal sample was transplanted into 3 mice. And because we had 6 fecal donors for each group, that leaves 18 mice that were colonized with human fecal samples per group. So these 2 groups were matched for sex. And then we didn't see differences in body weight or in body composition. And this was actually a surprise because whereas in the larger human group, we saw differences of about 5% for whole body lean mass and fat mass, the human fecal donors were actually more extreme than that. There were differences of 10%. So the high functioning human fecal donors had 10% more lean mass than the low functioning fecal donors. So, but nonetheless, it didn't transfer in this study. And then when we ran the animals to exhaustion on a treadmill, we didn't see a significant difference. And then you could even make an argument that the trend would be going towards the opposite direction for the low functioning colonized mice to have better endurance capacity. And I could potentially talk about why that would be, even though I should mention it's close to significance, but not significant. But then interestingly, we actually did see a difference for grip strength. So the high functioning colonized mice had a small but significant increase in grip strength. So it's shown here. So based on these data, because of the overlap for those various bacterial taxa that we saw, are they related to mechanisms involved in muscle strength in older adults? So what would next steps from this be? So if we transplant those bacteria into germ-free mice, would we see changes in muscle strength? And if so, how? Privitellaceae and Barnesiella contain genes that produce these short-chain fatty acids. Are they involved? And then conversely, if resistance training, RT, is well known to improve muscle-related measures, including muscle strength. So if we improve muscle strength with resistance training, does that affect the gut? And if so, which bacteria change as a result? So these are my next steps to be pitched in grant submissions coming up. So going further from here, earlier this year, our group published the potential role of diet and dietary fiber on the gut-muscle axis. So earlier, I presented data that short-chain fatty acids positively impact muscle, and dietary fiber isn't homogeneous, meaning it's comprised of insoluble fiber, which is not fermented by gut bacteria, and soluble fiber, which is fermented by gut bacteria to produce short-chain fatty acids. So how is dietary fiber related to the gut-muscle axis? Very few studies have looked at this. Only one RCT has been performed. And in that study, they looked at the change in grip strength in response to 13 weeks of soluble fiber supplementation. In this case, it was inulin and fructooligosaccharides. So 13-week supplementation in older adults. And what they found was, looking at grip strength of the right hand and the dominant hand in this cohort, they saw about a 20% increase for the soluble fiber supplemented group when compared with baseline, whereas the placebo group actually saw a decrease in their grip strength over the 13-week study. Now, I didn't mention it, but they did not see differences in the grip strength of the left hand when for either the placebo or the soluble fiber supplementation. So to date, this is the only study that's looked at it. So more study is needed to investigate if dietary fiber can alter muscle-related outcomes. So it's also important to note in this study, even though they gave soluble fiber as a supplement, they didn't look at changes in the microbiome or short-chain fatty acids. So how this could even occur, it would be unknown. So to further explore the role of dietary fiber on the gut-muscle axis, I recently analyzed data from a large study, the osteoporotic fractures in men study, also known as Mr. Oss. And this is unpublished data. We should have put it in the paper, but we didn't, but nonetheless. So what we're looking at here is the percentage of whole body lean mass, and on the x-axis, fiber density. So this is grams of fiber normalized to 100 calories. So if you just look at fiber intake versus lean mass or any outcome measure, you know, one could argue that with higher fiber, there could also be higher calories. So by looking at fiber density, you kind of account for that. You're accounting for calorie intake in addition to fiber intake. So what we see is that there's an increase, significant increase for fiber density with the percentage of whole body lean mass. So as fiber density of the diet increases, there's an increase for the percentage of whole body lean mass. So to further investigate this in terms of the microbiome and its functions that could be playing a role, we decided to separate that data into tertiles. So a group that was in the highest tertile for dietary fiber density and the percentage of whole body lean mass, and then the lowest tertiles for those two variables. So our cohorts, we had 42 in the high fiber density, high lean mass group versus 32 in the low group. These were 85-year-old men. Women were not included into the study, unfortunately, to look at the effect for women if it was similar. And then other key points here are as expected, the high fiber, high lean mass group had about 13% better or higher whole body lean mass and about 13% lower whole body fat mass. So a better body composition and lower BMI was found in the T3 or high fiber density, high lean mass, percentage of lean mass group. But they also were more physically active. So we accounted for that by adjusting all of our microbiome models for their physical activity score. It's also important to note that this group, the high fiber, high percentage lean mass group, also had better physical function, so higher short physical performance battery score and better grip strength. And then, as I noted, they had a higher dietary fiber and a higher fiber density when compared with the low fiber, low lean mass percentage group. So essentially, we have a group that's got better physical function and a better body composition, and that consume more dietary fiber. So what did we see for changes in bacteria, gut bacteria and function? So this is a genus level plot visualization. So the high fiber, high whole body lean mass group versus comparatively the low group. And just to highlight a couple key findings. So ruminococcus were lower in the low fiber group and higher, you can see that here, the orange, lots more orange versus the low group. And also I've illustrated here, tabular format. And actually by percent relative abundance, the differences from ruminococcus were greatest in terms of how much of them were there in the high fiber group. And then conversely, Parabacteroides were higher in the low fiber, low lean mass group when compared with the high fiber group, which had less. And again, we can see that here. Now, what we can't see here because these plots are difficult to read when you have much lower relative abundance, but Clostridium and Lachnospira were also higher in the high fiber group. And these three, Clostridium, Ruminococcus, and Lachnospira, are known butyrate-producing bacteria. So investigating further, we also looked at gut bacterial functions. And without going into that data, but just noting the key findings, we saw that the genes that produce butyrate were higher in the high fiber, high percentage of lean mass, high physical function group when compared with the lower group. So while these associations are interesting, and it suggests a role for dietary fiber on the gut muscle axis in older adults, they're just associations. So in a couple of grants so far this year, I've pitched this idea that a high soluble fiber dietary intervention, and coming from whole food and not from supplemental soluble fiber, and I can get into that, the why behind that. So a high soluble fiber dietary intervention may positively affect the gut muscle axis, and maybe even through short chain fatty acids and other related metabolites. So future study. So that's all I've got. Ready for questions. And if you guys want to find me, anyone wants to find me, I've got lots of, you can find me in a lot of places. So thanks a lot.